ठीक है. So that was the last thing we have done. हमने देखा था यहाँ पे कि हमारे यहाँ पे किस तरीके से एनाफेज यहाँ पे हो रहा है. एनाफेज के पहले हमने यहाँ पे क्या क्या देख लिया था. हमने देखा था हमारे यहाँ पे चेक पॉइंट नंबर 3 क्या करता है, हमारे यहाँ पे चेक पॉइंट नंबर 2 क्या करता है, 1 क्या करता है. So that is something we have seen. तो क्या आप में से कोई मुझे बता सकता है, कैन एनीवन ऑफ यू ओवर हियर टेल मी व्हाट योर चेक पॉइंट नंबर 3 वाज नोन फॉर? व्हाट इज दिस चेक पॉइंट नंबर 3 इज एक्चुअली गोइंग टू डू? कैन एनीवन टेल मी दैट? व्हाट इज दैट गोइंग टू बी डूइंग? हां शिखा बताइए, व्हाट दिस चेक पॉइंट नंबर 3 इज नोन फॉर?
सो ये बेसिकली चेक करता है कि स्पिंडल फाइबर्स करेक्टली अटैच है या नहीं है उससे काइनेटोकोर से.
ठीक है, right, that is what it does, right? So if they are not correctly attached, then you're going to ensure that they are being very correctly or very firmly attached to the kinetochore. True, very true. And along with that, once they are correctly attached, once you know that my kinetochore are very firmly attached, then uske baad you start your anaphase with the help of my anaphase promoting factor complex. So APCR written, APC is my anaphase promoting complex. With the help of that, you actually promote your anaphase. That's when you will find the anaphase is going to initiate. So this is something you should be familiar with, that anaphase protein complex is the protein complex which actually gives you the last signal कि अब चलो चालू करते मेरा anaphase. ठीक है, तो ये आपका होता है और इसके फिर आपका anaphase यहाँ पे चालू होगा. As soon as the anaphase is going to start, what will happen, that you will find the centromere is going to break. As the centromere is going to break, what will happen, this chromatid will start moving towards the respective pole. ये जो मेरे chromatid है ये अपने अपने poles की तरफ यहाँ पे जाना चालू करेंगे. So they will start moving towards the respective pole. So याद रखना, anaphase can be always remembered as a event which has been also in short going apart. So over here the chromatids will start going apart, centromere is going to break, and that is what you find happening in the anaphase. और कुछ यहाँ पे नहीं हो रहा. That's a very simple thing happening in the anaphase, and it's a very simple process. After anaphase, you will be having your telophase. So once you're done with the anaphase, now you will have the last stage of the mitosis. That is going to be the telophase. So let's quickly have a look at what are the events which will be happening in the telophase. Right. Now, the first thing you have to understand here is that your chromatid have reached to the respective poles. So आपके chromatid, they have reached towards the poles. ये chromatid जो मेरे है ये मेरे poles तक पहुंच चुके हैं. Okay. ये भी यहाँ पे पहुंच गए, ये भी यहाँ पे पहुंच गए. They have reached towards the poles. Now what will happen? These chromatids, once they are towards the pole, they will undergo decondensation. That means now they will lose their shape and they will turn into a thread-like DNA. So that's the first point you have to know, that your chromosomes undergo decondensation, and they will turn into chromatin. Now, if you remember, during the prophase, what was happening? Your chromatin was undergoing condensation to form the chromosome. Over here, what is happening? My chromosomes are undergoing decondensation. That means now they are beginning with a thread-like structure. They are forming a thread-like DNA. So it is something which is completely opposite to what was happening in the prophase. So आपका जो भी prophase में हो रहा था उससे बिल्कुल opposite आपके यहाँ पे हो रहा होता है. Right? Similarly, over here also this chromosome will also undergo decondensation to form into your thread-like DNA. So that is something you find happening over there. That's the first thing you have to know. Chromosome undergo decondensation to form the chromatin. That is one thing you have to know. After that, the next thing you will be finding happening here is your nuclear membrane is going to reappear. Your nuclear membrane is going to reappear. So you have a nuclear membrane, and that nuclear membrane is going to reappear. So that nuclear membrane is going to reappear. Again, that is something opposite to my prophase. So nuclear membrane reappears. That is also one thing you have to know. Your nuclear membrane going to reappear. That's the second thing you'll find happening in this. Along with that, you can very clearly see that your spindle fiber is no longer seen. So spindle fiber disassembles during the telophase. So spindle fiber आपको यहाँ पे दिख नहीं रहा. आप कह सकते हो कि वो disassemble हो गया या वो vanish हो गया. So my spindle fiber disassembles. Spindle fiber disassembles. मतलब spindle fiber is not something you can see now over here. So that is one thing you have to know. So my chromosome undergo decondensation to form chromatin. Nuclear membrane आ गई मेरे यहाँ पर, spindle fiber यहाँ पे आपको दिख नहीं रहा. You cannot see spindle fiber over here, so it has been lost, right? So these are the three important events you find happening in the telophase. And along with that, you can remember one more event, that is nucleolus reappears. or I would say all the organelles and the nucleolus are going to reappear. So you'll find my nucleolus is going to reappear. Nucleolus reappear. Along with that, all the organelles like your mitochondria, Golgi, ER, which cannot be seen right now, they will start, you will be able to observe them. Okay. So these are the events which have been happening during my particular telophase. Now the next thing you have to know is going to be, तो ये तो आपका हो गया. So my nuclear division has taken place. My two nucleus are being seen over here, but it is still a single cell. ये भी आपका single cell ही है. ये भी आपका single cell ही है. तो आपको यहाँ पे क्या मिलेगा? इस single cell के अंदर आपको ऐसे furrow formation आपको मिलेगा. You'll find such furrow formation will be seen. So it is still a single cell, but in this single cell, you'll find furrow formation will be observed. So this is called as my furrow formation. So furrow formation, this furrow formation will be seen happening in the cell. Now, when you find this kind of furrow formation happening, that actually means what? That the cell is now going to prepare itself for the cytokinesis. We are done with the karyokinesis. Your nuclear division is called as my karyokinesis. So आपका nuclear का division आप यहाँ पे हो चुका है, आपके दो nucleus अलग बन चुके हैं. Now you have to carry out my cytokinesis, and to carry out cytokinesis, you need to have this furrow formation. So this furrow formation will be seen happening over here. यहाँ पे भी आपको furrow दिख रहा है, यहाँ पे आपको furrow दिख रहा है. And now this furrow will start going into the much more in the center area. It will go towards the center side, or in towards the center it will start moving inwards, and then you will find ultimately the two new cells will be formed. So how this is going to look like? So initially you will have a cell which will have this kind of like furrow formation will be seen over there. Okay, and you will find slowly this furrow will start moving towards the inner side. This furrow will keep on moving towards the inner side, and in a matter of some time, what you will get to see that these two cells will get separated from each other. So now two cells, ये आपका furrow center में आ जाएगा एकदम, और एक बार आपका furrow यहाँ पे center में आ गया, then you will find two new cells will be formed, right? Let me make a nucleus over here so that you can see the nucleus has also been seen over here. So you have a nucleus over here, you have a nucleus over here as well, right? So then you will find. So this is something what you call as a cytokinesis. So you have to be very clear with this. Initially, we carry out the karyokinesis. That means the nuclear division will take place over there. And after that, you will find my cytokinesis will take place. उसके बाद मेरे यहाँ पे cytokinesis होते हुए मुझे यहाँ पे दिख जाएगा. और cytokinesis होने के बाद आपके यहाँ पे दो सेपरेट सेल आपके यहाँ पे आपको बन के मिल जाएंगे. So you find your two separate cells are being formed. This is what you find happening in the mitosis. तो याद रखनी है आपको ये बात कि टेलोफेज के अंदर क्या क्या हो रहा है. क्रोमैटिन फॉर्मेशन मेरा यहाँ पे हो रहा है. न्यूक्लियर मेम्ब्रेन मेरी वापस आ रही है. The nuclear membrane is again coming back. My spindle fiber disappears, right? My nucleus also reappears over here. So nucleus is also something which can be seen. The way nuclear membrane is coming, nucleus can also be seen coming over here, right? Now, when you find these events happening, they are events which are completely opposite to the prophase. अगर आप events को अगर आप देखते हो यहाँ पे तो आप समझ पाओगे ये जो मेरे events यहाँ पे ये बिल्कुल opposite है मेरे prophase से. Prophase के अंदर जो भी हो रहा था उसके बिल्कुल उल्टा यहाँ पे आपको होते हुए आपको यहाँ पे मिल जाएगा. ठीक है? Completely opposite things will be seen happening over here, and that is the thing you see happening in my telophase. So अगर if they ask you a question, which phase of mitosis is going to be completely opposite to my prophase, then the answer is going to be my telophase. Telophase happens to be completely opposite to your prophase. Okay, and you furrow formation will be seen happening. Now this furrow formation is only seen happening in my animal cell. Yaad rakhna, this furrow formation is always seen to be happening in my animal cell. So yaad rakhna, this should be clear to you that furrow formation is only seen to be happening in my animal cell. This is something you find happening in your animal cell. Right. Now you don't find this happening in the plant cell because plant cell have a cell wall and cell wall is very rigid. So please remember this thing. This furrow formation, you can find that happening in the animal cell, but you don't find them happening in the plant cell because plant cell is very rigid. It has a strong cell wall. And because of that, you don't find any kind of like furrow formation happening over there. ये बात याद रखना. ये बात कई बार आपके क्वेश्चन में आ जाती है. Very often you find this coming as one of the question that cell wall is not extensible. cell wall in the plant cell is not going to be extensible because it's a cell wall which is a hard structure. So you don't find cytokinesis happening in plant cell like this. Cytokinesis takes place in the plant cell in a certain way, in some other way, right? We'll see that other way later on. तो इतनी बात आपको यहाँ पे याद रखनी है. These are the few questions we can have a look at quickly. So we are done with the mitosis. We are done with the mitosis. We have started with the prophase. हाँ जी मिसबा बताइए. अच्छा अच्छा आंसर बताना है. Okay. Okay. Yeah. Misbah, tell me what will be the answer of the question by 18.
Also, I want to
हाँ
say
बोलो
repeat once again.
बोलो बोलो सवाल पूछो बताओ.
Please repeat once again.
Which part? This part.
Yes, sir.
So what will be happening here in the Misbah that you will get to see that you have understood the cytokinesis, right?
Yes, sir.
So what happens now? This what is happening? This cell membrane going inside is called as my furrow-formation. This is called as my furrow-formation. Something I've already told you. Now this cell membrane going inside is not something you will find happening in my plant cell. ये plant cell में नहीं होता. You don't find this happening in the plant cell. Now the question is why it does not happen in the plant cell. The answer is in the plant cell furrow formation won't be happening because plants have a cell wall and cell wall is very rigid. So that is why in them cytokinesis will take place in a different manner. So in them the cytokinesis will take place, but it will have another way of dividing the two cytoplasm. Okay, not like the animal cell. So ये आपको मिलेगा होते हुए आपके animal cell के अंदर, not in the plant cell. Misbah, is this clear to you?
Yes, sir.
ठीक है सर. हाँ जी बताइए महालक्ष्मी. महालक्ष्मी, first of all, tell me why you are late in the class.
सर मेरा फोन मेरे छोटा भाई के पास था.
हाँ तो ले देखा ना फिर उससे क्लास का टाइम आने के टाइम
अभी
पे.
दिया सर.
ऐसे कैसे चलेगा? ठीक है, बताओ महालक्ष्मी क्या कहना चाहते हो, बोलो.
सर इन एनिमल सेल से फरो फॉर्मेशन होगा सर. इन प्लांट सेल में मतलब एक प्लेट होता है ना सर, सेल प्लेट से डिवाइड होता है.
सेल प्लेट फॉर्मेशन, हाँ तो हम बाद में देखेंगे कि वो सेल प्लेट फॉर्मेशन से कैसे होगा. ठीक है. सो या सो इट हैज अ डिफरेंट वे ऑफ कैरिंग आउट द साइटोकाइनेसिस. ठीक है. तो प्लांट सेल का डिवीजन हम लोग बाद में देखते हैं. हाउ द साइटोकाइनेसिस टेक्स इन द प्लांट सेल वी डू आफ्टर द मियोसिस. ठीक है. चलो, सो आई विल जस्ट पिक अप फ्यू क्वेश्चंस ओवर हियर. नाउ यू हैव टू टेल मी क्वेश्चन नंबर 18 का आंसर यहाँ पे क्या आएगा. नाउ बताओ जल्दी. महालक्ष्मी बताओ क्या होगा इसका आंसर.
काइनेटोकोर सर.
एबीसीडी बताओ. एबीसीडी. स्पिंडल फाइबर अटैच टू व्हाट? एबीसीडी कहाँ पे है? ऑप्शन ए. आप
ऑप्शन सर ऑप्शन डी.
गायत्री क्या लग रहा है?
Sir, option D.
Option D is going to be the correct answer. Always spindle fiber attaches to the kinetochore of the chromosome. It always attaches to the kinetochore of the chromosome. Please don't miss out. That is where you will find a spindle fiber will get themselves attached. ये वाला क्वेश्चन भी आप कर सकते हो. Anaphase promoting complex is a protein degrading machinery necessary for the proper mitosis. I have already told you what is my APC. Something I have already explained. If of an animal cell, if a APC is defective, अगर आपका APC काम नहीं कर रहा, if anaphase protein complex is defective in the human cell, which of the following is expected to occur? तो क्या होगा आपके सेल के अंदर अगर APC काम नहीं करेगा तो? बताइए मिसबा.
Option D.
ऑप्शन डी. आर यू सेइंग ऑप्शन डी या ऑप्शन बी? क्या कह रहे हो आप?
Option B.
बैटमैन. अच्छा. या टेल मी शिखा.
सर ऑप्शन बी. chromosomes will not segregate.
Chromosomes will not segregate. That is going to be a correct answer. Yes, your chromosomes are not going to segregate if you have your APC is defective, right? They will not segregate. So yes, option B becomes the correct answer. At metaphase, chromosomes are attached to spindle fiber. This is something we have done. This will be having your options. It will be the correct answer in this. Something we have done this, right? Now, this is a very good question. If you are provided with the root tip of onion in your class and asked to count the number of chromosomes, or बोला कि count करो chromosomes को, which of the following stages can you most conveniently look into? Out of these 4 stages, which of the stage you can conveniently see for the counting the chromosome? Again, it's a simple question. Gayatri, what do you think? What will be the answer over here?
The metaphase.
All right. Aisha, what do you think? Aisha is not there in the class. Yeah. Okay. It is going to be your metaphase. Metaphase आपके यहाँ पे science होगा. Metaphase is the best time. If you want to count a chromosome, if you want to study chromosome, metaphase is the best time for you. Right? Microtubules means your something which is coming out from the. Tell me. Question number 32 का आंसर क्या होगा? Hand raise करो 32 के लिए. Microtubule is involved in. Let me give you a hint. Microtubule are coming out from the centrioles. Microtubule are coming out from the centrioles. बताइए महालक्ष्मी. Can't hear your answer. Microtubules are involved in. See, microtubules are coming out from the centrioles. क्या कर रहे हैं आप लोग? It will come out from the centrioles. अगर ये centriole से आ रहा है तो समझ जाओ ना इसका आंसर क्या होगा. It is going to be playing the role.
Cell division.
Cell division. Right. It will have a role in my cell division. Right. ठीक है. अब ये बताओ. During cell division in the apical meristem, the nuclear membrane appears. Right. So nuclear membrane वापस कब आती है? देखो क्या लिखा है इन लोगों ने. Nuclear membrane appears. Now you have to tell me directly बता दो कि nuclear membrane वापस कब आती है? कौन से फेज में आपके आती है?
Telophase.
Telophase.
Right. So telophase will be the correct answer. हाँ जी, आंसर है. Telophase is the one जहां पे आपकी nuclear membrane आपकी वापस आ जाती है. How many mitotic division are needed for a single cell to make 128 cell? Let me help you. इसका एक रूल याद रखना. 2 raised to n. 2 raised to n. So n जो होता है ना, it is my number of division. n मतलब number of division. तो अगर मैं करता हूँ यहाँ पे 2 raised to 1, so that means 2 raised to 1 क्या आता है मेरा इसका आंसर? It is going to be 2. Right? तो एक डिवीजन के बाद मुझे मिलेंगे मेरे यहाँ पे 2 सेल. Right? Similarly, just wait a minute. Similarly, what will happen here is that if I write over here 2 raised to 2, तो इसका आंसर क्या होगा? 4. राइट. सो 2 डिवीजन के बाद ये मिलते हैं 4 सेल. राइट. तो उसी तरीके से आपको अगर पूछा गया कि 128 सेल कब मिलेंगे आपको, तो इसका अगर आप इसको निकालोगे इसका आंसर, सो व्हाट विल हैपन, ऊपर रेस टू आपका आएगा यहाँ पे 7. 2 raised to 7 जब आप कैलकुलेट करोगे तो यू विल हैव आंसर दैट्स गोइंग टू बी योर 128. सो 7 बार जब डिवीजन होगा सेल का तब जाके 128 सेल मिलेंगे. तो याद रखना ये 2 raised to n वाला फॉर्मूला याद रखना. फॉर्मूला मतलब एक, या दिस इज समथिंग यू हैव टू रिमेंबर. सो n होता है आपका नंबर ऑफ डिवीजन और उससे आपको मल्टीप्लाई करके देख लेना है कि आपका क्या आंसर निकल के आ रहा है. सो आफ्टर 7 डिविजन्स यू विल हैव दिस 128 सेल. Now let's do this question. ये सारे simple question है. हाँ, cells at the end of prophase when viewed under the microscope do not show. Cells को जब आप observe करते हो prophase के अंदर, by the end of the prophase आपको क्या चीजें नहीं दिखती? What are the things you don't get to see? Do not show. What are the things you don't see over here? Raise your hand to answer this. Gayatri, tell us.
Sir, all of these.
Answer.
All of these is going to be a correct answer. You cannot see any of these things. You cannot see Golgi, ER, nucleus. Everything is gayab. By the end of the prophase, you don't get to see any of those things. So yes, option D becomes the correct answer. Some question which I'm leaving right now is something you can do as a homework. Okay, they are very simple questions. Metaphase is not characterized by. Which of these events are not seen in metaphase? Again, it's a very simple question. You just have to put some logic over here. Shikha, क्या होगा इसका आंसर? Metaphase is not characterized by.
Sir, option A.
Read again. Read all the options again.
Answer D.
हाँ, right. Option D आपका जो आएगा वो आपके यहाँ पे सही आंसर, because option D is completely different. Splitting of chromosome is never something which is can be seen happening in the metaphase. Okay, splitting of chromosome can only be seen happening in my anaphase. So that is why option D over here becomes a completely odd one out as a correct answer. Okay. सो पूछा यही था कि कौन सा इसमें से ट्रेट मेटाफेज में होते हुए आपको नहीं मिलता. सो ऑप्शन डी ओवर हियर बिकम्स माय करेक्ट आंसर. राइट. एनाफेज इज कैरेक्टराइज्ड बाय. ऐसे ही बता दो, अनम्यूट करके बता दो इसका आंसर क्या होगा. एनाफेज इज कैरेक्टराइज्ड बाय, वैसे बता दो.
सर ऑल ऑफ दिस.
सर ऑप्शन डी.
हाँ
Option D.
राइट, सबका आंसर यहाँ पे सही है. ऑप्शन डी इज करेक्ट आंसर. ये सारे इवेंट्स मुझे एनाफेज में होते हुए मुझे वहां पे मिल जाते हैं. ठीक है. Alright, so these are the questions. You can have a look at them later now. Now let's have a look at the meiosis. One of the very important event of the chapter is going to be your meiosis. So now when you get a term like a meiosis, first thing you have to know that meiosis is something which is being done for gamete formation. Right? This is something which happens during the gamete formation. So when the gamete has to be formed, then you will find the meiosis will take place. The second thing you have to understand here is that meiosis is something which takes place in the sexually reproducing animals only. So during the sexual reproduction, this meiosis will take place, right? So during sexual reproduction, you will find this meiosis is going to take place. So you will have a gamete formation happening during the meiosis, right? So meiosis is an event through which you will be finding the gametes have been formed. So all your male cells, male gametes or female gametes, whenever they are been formed, they are formed because of the meiosis. वो meiosis करके ही बनाया जाता है, right? And also remember, it is my reduction division. It is also been called as my reduction division. Now these are some of the basic things you should know. They are called as my reduction division, right? Now something you have seen, we have discussed the meiosis when we were discussing the plant kingdom. If you remember, हमारे एक सेल में से 4 सेल बनते थे. If you remember, we used to make those cells. We used to have this single cell, diploid cell, and from the diploid cell, we used to make those 4
Haploid.
cells, right? Yeah, we used to make those 4 haploid cells. So that was something which was a meiosis, right? Now on the right hand side, you can see a picture over here where it has been very clearly shown that you have a diploid cell. From that diploid cell, you will have these 2 cells which have been formed initially, and then from these 2 haploid cell, again the 2 more cells are been formed. So that is how from a single cell, 4 haploid cells are been formed. Okay, so इसको हम बोलेंगे यहाँ पे as a meiosis 1. This is my meiosis 1 over here, and after that, this is my meiosis 2. So meiosis will have 2 parts. याद रखना, meiosis will have 2 parts. It will have a meiosis 1. It will have a meiosis 2. Meiosis is always been done during the gamete formation. During sexual mode of reproduction, you have to form the gametes. They have been formed via meiosis, and it is a reduction division. So here the number of chromosomes are getting decreased. आपका वो डिप्लॉइड था, आपका यहाँ पे हैप्लॉइड आपको गैमेट मिल रहा है, राइट? एंड याद रखना,
Yes.
हमेशा आपके डिप्लॉइड सेल में ही होगा. ठीक है. कोई, आई होप इसको लेके कोई कंफ्यूजन नहीं है. आई होप यू हैव नो कंफ्यूजन ओवर हियर. इफ यू हैव एनी कंफ्यूजन, देन प्लीज लेट मी नो. Right. Now let's have a look over here. You can see a mitosis takes place in the somatic cell. Mitosis always takes place in the somatic cell, right? A diploid cell will undergo mitosis to give you one more diploid cell, and they are identical to each other. याद रखना, जब mitosis आपका होता है, so whatever the cells you're getting, they are going to be identical to each other. So just wait a minute. By the way, आवाज आ रही है आप लोगों को मेरी यहाँ पे clearly?
Yes, sir.
नेटवर्क का तो कोई कंसर्न नहीं है ना, बिकॉज एट टाइम्स इट हैपेंस ओवर हियर. अभी ओकेशनली आज हुआ था. सो जस्ट लेट मी नो इफ एट ऑल दैट इज अ केस. आवाज अगर कटने लगे तो बता देना. दे आर गोइंग टू बी आइडेंटिकल. बिल्कुल डिक्टो होता है यहाँ पे काम. ठीक है, माइटोसिस के अंदर. लेकिन जब मियोसिस आपका होता है, you get 4 haploid cells, and these 4 haploid cells are going to be different from each other. चारों haploid cell एक दूसरे से अलग होंगे और अपने पेरेंट से भी ये लोग अलग होंगे. All the 4 haploid cell you will get to see over here are going to be different from each other, and they are also different from the parent cell. So that should be clear to all of you that this is what you find happening in the meiosis, that you get to have this 4 haploid cell. They are going to be different from each other, right? So this is something you have to know. Now, next thing. So here you can see the meiosis will have a meiosis 1, you will have a meiosis 2. right? And then ultimately you will have your 4 haploid cells have been formed. Now, before the meiosis starts, you will have a cell cycle. The way you have a G1 phase, S phase, G2 phase, you will have the cell cycle, right? You will have an interphase. Now, during the interphase, you will have a S phase, and S phase is known for what? S phase में क्या होता है बता दो मुझे जल्दी. What is my S phase is known for?
DNA replication.
हाँ जी, S phase is known for my DNA replication. बिल्कुल सी बात है. S phase आपके यहाँ पे जहाँ जाता है for my DNA replication. So that is something which will take place in the S phase, and then you will have the G2 phase, and then you will have the meiosis. Please remember one thing very clearly. DNA replication takes place only once. Even though you have a meiosis 1, meiosis 2 happening, please ऐसा मत सोचना. Don't think that DNA replication will happen twice. It will only take place once in the S phase. That's it. ये बात भूलना मत. DNA replication takes place only once. So this should be very, very, very clear to you by any means. Right? चलो then आगे बढ़ते हैं. तो ये वही आपके सामने पिक्चर है यहाँ पे. You can see your meiosis taking place. Now let's start with the, okay, now let's understand one thing first of all over here. When we discuss meiosis, meiosis itself is being divided into like meiosis 1, meiosis 2. Now let's understand, let's start with, obviously we'll have to start with the meiosis 1. Now when we discuss meiosis 1, what is been seen happening in this, you have like 4 stages. You'll have your prophase 1, then you will have your metaphase 1. अच्छा, बाय द वे आप लोग की स्कूल की छुट्टियां कब से चालू हो रही है? वेकेशन, स्कूल वेकेशन, समर वेकेशन.
सर परसों से.
शिखा का परसों से हो रहा है. मिसबा आपका चालू ही है? अच्छा, स्टार्ट ही है. ओके. गायत्री
अभी
योर,
कंफर्म नहीं है, मतलब एक्स्ट्रा क्लासेस हो सकते हैं.
अच्छा, वो तो ड्रॉप कर सकते हैं ना एक्स्ट्रा क्लास. वो छोड़ नहीं सकते एक्स्ट्रा क्लासेस. कांट यू लीव दो एक्स्ट्रा क्लासेस? जाना जरूरी होता है.
यस सर.
अरे शिखा, जाने दो एक्स्ट्रा क्लासेस, कौन जाता है? हाँ गायत्री, योर समर वेकेशन.
Sir, already started, sir.
And till what date you have this vacation, Gayatri?
Sir, June 1, sir.
June 1. Okay. Anyone else in the class? We have like Gayatri is there. Okay. Aisha, summer vacation till when?
This 14th
Till 14th of June.
May.
Acha, it will start on 14th of May. All right. Okay. Okay. So you have this meiosis 1, and it will have like 4 stages. You have a prophase 1, metaphase 1, anaphase 1, telophase 1, and then after that you will have a meiosis 2. Now our focus is to understand the prophase 1. Our main focus over here is going to understand my prophase 1, and then we can see the remaining shape. Because if you understand the prophase 1, the rest things are like very simple. Okay. So now let's have a look over here. Something in terms of the prophase 1. Let's see what will be happening over here. Now, so our focus will be right now is going to be my prophase 1, because it is one of the most important event of my meiosis. Right? बाकी सब तो ठीक है, prophase 1 पे हम focus करेंगे. Now, prophase 1 itself will be divided into like several stages. Prophase 1 will itself be divided into like several stages. So now let's try to see them. So when we discuss prophase 1, it will have certain stages like, first of all, it will have my liptotin stage. Then it will have the second stage will be my zygotin stage. This is to be remembered. You have a liptotin, then you have a zygotin, then you have a packitine. Then after that, you will have your diplotin. And then you will have something like your dikinesis. This should be remembered. First of all, that prophase 1 itself is been divided into like 5 other stages. We have a liptotin, zygotin, packitine, and your diplotin. And then you have a dikinesis. So these are the 5 stages of my prophase which has to be always remembered. ये आपको याद रखना है और इनको ही आज हमें डिटेल में समझना है. If you are able to understand this much amount of thing, then this is the main part of the chapter. You should be able to crack most of the questions. So yeah, definitely sequence. Please remember the sequence. It starts with liptotin, zygotin, pyridine, liptotin, and diagenesis. So if someone asks you, if one of your friend asks you that which part of my meiosis is the longest one, then the answer is going to be very simple. It is my prophase 1. Prophase 1 happens to be one of the longest phase of the meiosis, right? So that should be clear to all of you. Okay. यहाँ तक सब क्लियर है? यहाँ तक से समझ आ रहा है? Is this thing clear to all of you?
Yes, sir.
All right. Okay.
Yes, sir.
Okay.
Yes,
Now
sir.
want to ठीक है. So there's something we'll get to see. Now let's start with my liptotin stage. Let's see what is it. So understand, liptotin is a part of my prophase. It is a part of my prophase 1. So let's start with my liptotin stage. Now, what do you get to see happening over here? So some very simple events will be happening over here. First of all, you will find my chromatin will condense to form chromosomes. My chromatin will get condensed to form chromosome. Right? That's one thing you'll be finding happening. That means you will have your thread-like DNA. It will get condensed and will turn into a chromosome. This is what you find happening in the liptotin stage. Second thing you have to know that my centriole will start moving towards the poles. My centrioles will start moving towards pole. That is the second thing you have to know, that my centrioles जो वो आपके होते हैं ना, वो आपके पोल्स की तरफ जाना चालू करेंगे. These are the only two things you have to remember in this. That's it. यहाँ पे आपका liptotin खत्म हो गया. बहुत short है आपका यहाँ पे, ये लंबा है ही नहीं है. That's my liptotin stage. My chromatin condenses to form chromosome, and my centriole will start moving towards the poles. These are the two things you have to know in this, right? So these are the things that have been mentioned over here. Chromatin condenses to form chromosome. Centriole begins to move towards the respective poles. That's it. खत्म हो गया ये. Now let's jump onto the second stage. क्या था second stage यहाँ पे? After liptotin क्या था दूसरा?
Zygotin.
Yeah, zygotin stage. So next one is my zygotin stage. Now understand what is happening in the zygotin stage. Zygotin is known for chromosomal pairing. So zygotin stage is known for my chromosomal pairing. So now let's understand what has been happening over here. ठीक है, ये ध्यान देना यहाँ पे. You will find even the exam which happened on Sunday, na, you had number of questions coming from this particular part. So you have a zygotin stage over here, and zygotin stage will have, it is known for my chromosomal pairing. Now let's try to understand what do you mean by chromosomal pairing. So assume that you have a chromosome number 7. Assume that this is my chromosome number 7. So it will come along with another chromosome number 7, because you always have chromosome in pairs. So it will come along with another chromosome and will get attached to another chromosome. तो ये chromosome 7 जो है वो उसके pair के साथ it will get itself attached with another pair, with its copy basically. This chromosome number 7 will get itself attached with its copy. So that is called as my chromosomal pairing, right? So 7 will attach with 7. Similarly, you will also you will have something like my chromosome number 9 or some other number, right? So all chromosome will start forming this homologous pair. So assume that you have one more chromosome over here, right? So it will come closer to its copy and will get itself attached. Now the question is, so this is something which will be happening over here. Chromosome will attach itself with its copy, right? Now the question is, attach kaise ho raha hai? How they are getting attached over here? That is the question over here. So pairing is happening, and after pairing, these chromosomes are actually forming an attachment with each other. So how that attachment is happening? So to make that attachment happen, there will be an involvement of some protein complex. So there is some protein complex which has been involved over here, and because with the help of that protein complex, this homologous pairing is being taking place. So you have some protein complex over here, and because of that, this chromosomal pair is getting itself attached with each other. So ये जो मेरा protein complex मेरा यहाँ पे है, इसको हम बोलेंगे, this has been called as my synaptinomal complex. Synaptinomal complex. So if someone asks you, अगर कोई शिखा को रात को भी उठा के पूछता है कि क्या होता है synaptinomal complex, then she should be able to tell that person that it is a complex which actually helps in attachment of my two chromosomes. It actually helps in attachment of my homologous pair. So my homologous pair is getting itself attached to each other with the help of my synaptinomal complex. That should be clear to you. It is one type of my nucleoprotein. This synaptinomal complex is a one type of my nucleoprotein. ये एक type हमारे यहाँ पे nucleoprotein है. It is one type of my nucleoprotein which actually helps in the binding of my two chromosomes over here. Right? This is also called as my, it has been also called as my zipper protein. You know what zip does, na? It attaches, like, you know what zip does. It is attachment of two like sides. So this is also called as my zipper protein. So this should be clear to all of you, right? This is what my synaptinomal complex is known for. Now, once this homologous pairing has been done, once you find my two chromosomes coming together and attaching themselves with each other, once this homologous pairing has been done, you will say one thing, that my synapses has taken place. Once this pairing has been done, then you can very clearly say कि यहाँ पे क्या हुआ मेरा यहाँ पे, I have carried out my synapses. So यहाँ पे मेरा synapses हुआ. What has happened over here? I will find my synapses has taken place over here. Okay, synapsis, right? So remember one thing, once the homologous pairing has been done, you will clearly say that my synapses has taken place. So they will ask you a question that in which stage of prophase 1 synapses will take place. The answer is going to be my zygote stage. So zygote stage is a stage where we will be finding the synapses will take place. Synapses simply means pairing of my homologous chromosome. Pairing of my homologous chromosome is called as my synapses. I hope this is clear to all of you. यहाँ तक Shikha, Misbah,
Yes, sir.
Okay.
and Gayatri, right? And Mahalakshmi. I hope it's clear to all of you. So yeah, this is what you find happening over here. Synapses has taken place. Now, there's one more thing you have to understand. So this is my homologous pair, right? Let me make one more pair over here. Let me quickly make one more pair. Okay, so you have this one more pair I've drawn over here, right? Now, when you see a homologous pair over here, and if they ask you that you have a homologous pair, right? This is my homologous pair, and you have a name for that. They are all both attached to each other. Na? This attached homologous pair is called as my tetrad. This is something called as my tetrad. Now, why it is called as a tetrad? Na? Because you have 4 chromatid over here. In a pair, you will get to see how many chromatid? 4 chromatid. 1, 2, 3, 4. Total number of chromatid you can see over here are going to be 4. That's why you have a term for that. It is called as a tetrad. It has been also called as my bivalent. Bivalent means because you have a single pair will have 2 chromosomes. Every single homologous pair will have 2 chromosomes, and that is why you have one more term over here, and that term is called my bivalent. So please remember these 2 terms. You have a bivalent and a tetrad. Tetrad term is being used because you can see over here very clearly you have a presence of a 4 chromatid. And bivalent term is being used over here because you have a homologous pair, and this pair will have 2 chromosomes. This pair has 2 chromosomes, which you can see over here. That's why the bivalent term can be used. So you can get a question that bivalent and tetrad, where it can be seen. So it can also be seen in the zygote stage. So in the zygote stage, you can very clearly see the presence of the bivalent or a tetrad. So that should be again clear to you, right? Now let me just draw one larger one over here. Let me just draw one larger homologous pair. Right, so you have this chromosome and you have a copy over here. Right, so now both of them are going to be attached to each other, right? You can see both of them are going to be attached to each other with the help of the which protein? कौन सा था protein यहाँ पे? Synaptinomal complex, right? That was the protein which is going to make them attach over here. Now, if I ask you, so this structure which I've drawn over here is a bivalent, right? This structure which I've drawn over here is going to be a bivalent. This is a bivalent. It can be also called as my tetrad. It can be also called as my tetrad. Right? Now you have a tetrad or bivalent, whatever term I can use. Now if I ask you how many centromere have been seen in the bivalent, then your answer is going to be
2 on each chromosome.
say 2. 1 on each chromosome, total 2. If I ask you how many centromere are there, then the answer will be 2. एक यहाँ पे, एक यहाँ पे. In a single bivalent, you will have 2 centromere. याद रखना, you will always have 2 centromere in a single bivalent. क्या सबको ये बात समझ आ रही है? Is this thing clear to all of you? Gayatri, is this clear to you?
Yes, sir.
Same question goes to Aisha. Same question goes to Misbah and all of the other students over here. I hope it's clear to you, right? Yeah. So, yeah. So you will find, in a single bivalent, you'll have a presence of a 2 centromere. That's one thing you have to know. Second thing, what you have to understand here is that they will ask you that how many chromatids you have. So how many chromatids you have in a single bivalent?
4.
4.
Right. 4 chromatid. The number of chromatid you should know, you'll have 4 chromatid in the single bivalent. So that should be also clear to you. So this is something you have to always remember. This is something you always have to remember. They will ask you a question. And if they ask you where I get to see the bivalent and tetrad, then the answer is going to be my zygote stage. This is the stage where I get to see my bivalent and tetrad can be seen. Now, so this is what has been written over here. Zygote stage, 2 homologous chromosomes get attached to each other, right? Process of attachment of homologous chromosome is called as my synapses. So that is something you have to know. Process of attachment is called as my synapses. And the involvement of central complex will play the role. So this is a complex. Because of that, the attachment will be seen happening. So these are the 2 points should be clear to all of you. Now let's talk about my next stage. Next stage is going to be which stage? Batao.
Pectine.
Right. Next stage over here is going to be my packaging stage. Right. Now let's see what does actually takes place in the packaging stage. Now again, it's one of the most important stage or eventful stage of my meiosis, or I would say prophase 1. I would say meiosis ka sabse important stage is my packaging stage, because one of the very important event is happening over here. Right. Now let's see what actually happens. Let me draw a big large chromosome over here. So now you have another chromosome, and that chromosome, the green one, like you have a homologous pair, and this pair, I'm just drawing them with a different color so that we can create a difference between them. So you have one arm is crossing over with another arm, or I would say one arm of chromatid is crossing over with another chromatid. Right. So this is something which will be happening over here as well. So you have this arm over here will cross over with another arm. Okay. So this is something you have to understand over here. Now what is happening in this? So you have this homologous pair which we have drawn over there. We have this homologous pair, right? And you can see their chromatid are actually crossing their arms. The chromatid over here are crossing their arms. So what is happening here now? Why the crossing of arms is happening? Why one chromatid is crossing with another chromatid, or one arm is crossing with another arm? This is happening so that you can exchange the DNA over here. This is something which is happening so that you can exchange the DNA. So what will happen here now? The DNA on the green chromatid will get onto the yellow chromosome, yellow chromatid. So whatever DNA, a small amount of DNA from the green chromatid will get onto the yellow chromatid. A small amount of DNA from the yellow chromatid will get onto the green chromatid. A small amount of DNA from the yellow chromatid will get onto the green chromatid. Similarly, this thing will happen over there. So what is happening over here? You are doing actually here what? Exchange of genetic material. Genetic material over here is getting exchanged. So genetic material is getting exchanged between this chromatid. So between this chromatid, the genetic material is getting exchanged. So that is something you have to understand. So what is that? If someone asks you, someone gives you this picture and asks you, "Kya yahan pe ho kya raha hai? What is happening here?" Then you will tell here.
Crossing of arms.
Yeah. crossing over is taking place. Crossing over is taking place over here, right? And during this crossing over, what do you get to see happening now? What do you get to see happening? That your genetic material is getting exchanged. Your genetic material is getting exchanged. Is exchanged. This is what you find happening over here. That is one of the important events happening in the packaging stage. Now, before getting into this again, now there's something we have seen happening. Now, let me tell you one more thing. Let me tell you one more thing which will be seen happening in the packaging stage. पहले ये बताओ, इतनी बात यहाँ पे समझ आ रही है आपको? What is happening here?
Yes, sir.
अरे सब लोगों को yes बोलना है, और नहीं आ रहा तो बोलो if it's not clear. See, understand one thing. All of us are different from each other, right? We all of us are different from each other because of the variation, right? Because of variation, we are different from each other. We are not even different in terms of physical features. We are different even in terms of our like immune system, even in terms of like metabolism. हम लोग अलग हैं. किसका metabolism ज़्यादा होता है, किसका कम होता है, right? Forget human beings. I'll take an example of let's say example of a deer. You get into a forest. In the forest, you see 100 deers. Even though those deers appear similar to each other physically, they are different from each other. And why they are different from each other? Because of the event happening in the meiosis. Because of this event where the crossing over is taking place, the DNA is getting exchanged. Because of that, we have the variation in the population. इसके वजह से ही you get to see the variation in the population. Because of that, you have a prominent variation in the population. And that variation is a reason, because of that, a population is able to adapt with the new changing environment. हम सबके अंदर वेरिएशन है और इसकी वजह से हम लोग अडेप्ट कर पाते हैं हमारे एनवायरनमेंट के अंदर. आपने नोटिस किया होगा, you must have noticed that sometimes some disease happens to people very easily and some people do not get diseased, right? So assume that it's a month of COVID, right? So out of 100, some people will have a very heavy symptoms of COVID. Some people will have a very mild symptoms of COVID. Some people will have no COVID. So why that happens? Even though we are all human beings, why that is happening? Because we have a variation. All of us are different from each other. Some has a slightly stronger immune system. Some has a weaker immune system. Some has an immune system which does not allow any kind of disease like COVID. So that is something you have to understand. This all happens because of the variation in the population, and the variation comes because of this. Because of this crossing over, this variation will be taking place, right? Are you able to understand what I'm trying to tell you over here? Is this story clear to all of you? Aisha, Shikha, Misbah, and Gitanjali. Sorry, Gayatri.
Yes, sir.
Yes, sir. Yes.
Yes, sir.
Right? Okay. Please tell me if it is not clear, because it's one of very important event. Because of that, the population survives. इसके वजह से evolution हुआ है और इसकी वजह से सारी population survive कर पाती है. ठीक है? Now let me tell you before that one more thing I have to tell you over here. See, before crossing over, one thing happens. Let me divide the page over here. So before crossing over, so assume that these are my homologous pair. This is my one pair and iski copy. I'll make a different color over here just for a simpler understanding. Like we have its copy over here which has been attached. Now you have to understand one thing, that before crossing over, what will happen now? Some dense area will be seen in your particular synaptic complex. So you know what synaptic complex is known for. Now it is something is going to attach my two homologous pair over there. So you have this synaptic complex which is going to be attaching this homologous pair, right? Now what happens here now, on this synaptic complex area, you get to have some dense area will be seen. In this region, you will get to have presence of some dense area. So you can find some dense area in this region. Now the dense area which you find over here in this particular region, this dense area are called as my recombination nodules. These are some dense areas you can see under the microscope, and these dense areas are called as my recombination nodules. Recombination nodules. So that is the first thing you have to know. That let's say I give Aisha a slide. In that slide, she can see the two chromosomes are attached to each other, and there she can see some dense areas. अगर आयशा को वहाँ पे dense area दिखते तो क्या बोलेगी? That there is a presence of a recombination nodule over there. Now the question is, मैं उससे पूछूंगा कि यार ये recombination nodule में होता क्या है? then what her answer is going to be, that recombination nodule will have a presence of an enzyme. These areas will have a presence of an enzyme, and that enzyme is called as my recombinase. That enzyme is called as my recombinase. So that is also something you have to remember, that this dense area, which are called as my recombination nodules, will have a presence of an enzyme, and that enzyme is called as my recombinase. So recombinase enzyme is seen over there. Okay. Now let's understand what this enzyme is going to do. This is the enzyme which will help in the exchange of genetic material after crossing over. आपने यहाँ पे क्रॉसिंग ओवर कर लिया, ठीक है, उसके बाद जो आप डीएनए एक्सचेंज कर पा रहे हो, जेनेटिक मटेरियल जो आप यहाँ पे एक्सचेंज कर पा रहे हो क्रॉसिंग ओवर होने के बाद, दिस एक्सचेंज ऑफ जेनेटिक मटेरियल विच विल बी हैपनिंग आफ्टर क्रॉसिंग ओवर, ये करने के लिए आपका रिकॉम्बिनेज एंजाइम आपकी मदद करता है. दिस एंजाइम इज गोइंग टू हेल्प यू आउट in exchanging the genetic material. So this is something should be remembered, that this recombinase enzyme helps in exchange of genetic material after crossing over. Genetic material after crossing over. After crossing over. Once you're done with the crossing over, you need to exchange your DNA over there, and that will be done with the help of my recombinase enzyme. So this should be clear to you. So if they ask you in which stage of meiosis you get to see the recombinase enzyme working, your answer should be your packaging stage. If they ask you in which stage the crossing over will take place, the answer is going to be my packaging stage, right? So what actually happens? Crossing over will happen over there, right? You will exchange the genetic material, and once you have exchanged the genetic material, we will say recombination has taken place. आप बोलोगे यहाँ पे that there has been a recombination has been taken place. Recombination has taken place. एक बार आपने मटेरियल एक्सचेंज कर लिया ना, डीएनए वाला एक्सचेंज आपने कर लिया, देन आप बोलोगे यहाँ पे आपका हो चुका है, आपका यहाँ पे रिकॉम्बिनेशन. So please carefully remember, if they simply ask you, अगर कल उठ के आपका friend, if one of your friend from your own class ask you, where does the crossing over and recombination takes place, in which stage, then your answer is going to be my packaging stage. I know you don't have such kind of friend which are going to ask you this kind of question, right? कितना अच्छा होता है लेकिन अगर ऐसे friend होते तो, if all of you must be having would be having this kind of friends who ask you this kind of question, school में भी revision हो जाता, right? घर आके पढ़ना नहीं पड़ता. But yeah, so you have to know this thing, that packaging stage is a stage in which you will get to see your crossing over and recombination will be taking place. And when they tell you these are the events which are happening, then you should understand after crossing over, after you do the overlap, you have to exchange the DNA, and वो करने के लिए आपने यहाँ पे recombination enzyme आपकी मदद करेगा. This is the enzyme which will help you to make that DNA exchange takes place, right? So these are the events which you find happening in my packaging stage. This is one of the most important event, because of this event, what happens? You have a variation in the population. Because of this event, you have a variation in the population. Because of that, even though animals look similar to each other, they are different from each other because of this event. And this event is the reason, because of that the population survives. वरना अगर सोचो हर एक एनिमल सेम जैसा सेम होता, समझो हर ह्यूमन बीइंग का सेम होता है जेनेटिक मटेरियल, एक डिजीज होती ना, सब लोग जाते हैं. If all the human beings were similar, were identical to each other without any variation, what would have happened? A single disease would have wiped out a complete population of that area. So that is why you need a variation in the population, right? So that should be clear to all of you. Now, is this thing clear to all of you? Aisha, Ms. Gayatri,
Yes, sir.
and Kha. अच्छा, thumbs up दे रहे हो आप लोग? अच्छा अच्छा, ठीक है, right, okay. Sometime I was waiting, कोई बोलेगा. All right. So this is something you have to know, right? So soon after synapses, dense area appear known as a recombination nodule. It has an enzyme recombinase. Crossing over takes place over here. So this is where the crossing over takes place. Now there's one more thing I have to tell you. One more thing I have to tell you over here. Let me come back. Right. So I have drawn over here a bivalent. Now when I draw a bivalent over here, now remember a couple of terminologies over here. These two chromatids are called as my sister chromatids. These two chromatids are called as my sister chromatids. Okay. Similarly, these two chromatids of single chromosome are been called as my sister chromatids. So you know that what are my sister chromatids. right? But on the other hand, these two chromatids, these two chromatids, जहाँ पे crossing over होगा, these two chromatids are called as my non-sister chromatids. They are called as my non-sister chromatids. These is two sister chromatids are called my non-sister chromatids. So that is something should be clear to you. So these two chromatids over here are my non-sister chromatids. So now if I ask you, crossing over takes place between which, sister या non-sister chromatids? किसके बीच में होता है?
Non-sister chromatids.
Yes, crossing over will always take place between the non-sister chromatids. So याद रखना, these are my non-sister chromatids. Non-sister. Okay. Let me show you a couple of pictures which makes things easier for you to understand. So here you can see crossing over is taking place between the non-sister chromatids, and after that you have a recombination has been done. So crossing over bhi ho gaya yahan pe, and then you are finding the DNA has been exchanged. So that has successful recombination has been done over here, right? So over here you can see, over here you can see you have a homologous pair, you have a chromosome crossing over happening over here, and because of that you are finding the recombinants are being formed. The recombinants are being formed. So recombination has taken place over here, right? That is something you have to know. Now the next stage is my diplotin stage. Now let's see what will be the event happening in my diplotin stage. So now the story of my diplotin stage. Let's have a look. So when we discuss the diplotin stage now, what happens now? Now let me make again those chromosomes over here. Right, so this is my one chromosome and it has undergone crossing over with the green one. It has undergone crossing over, so you know it has undergone crossing over here. Right, now what happens now? Now tell me something quickly. These two chromosomes were attached to each other with the help of what?
Synaptomal protein.
Synaptomal complex. You can also call it in a simple word, zipper protein. Right? Yaad rakhna. These two chromosomes are attached with the help of the synaptomal complex. Now what exactly happens now? The synaptomal complex will start detaching or will undergo dissolution from the center. So from the center, synaptomal complex will now get itself detached or will become dissolution. So what we call it as that there will be a dissolution of synaptomal complex from the center. Center से chromosomes आपके free हो गए. From the center, what will be happening? The chromosomes will be now free from the center. So now the chromosomes are no longer attached from the center. They are free from here. So what has happened in this? You are finding there's going to be a dissolution of synaptomal complex. This is something you should remember. There's going to be a dissolution of synaptomal complex. So now it will not happen completely. What has been happening over here now from the center, now you can see both the chromosomes are free from the center. So now synaptomal complex will start breaking apart and my two chromosomes are like separated from each other from the center. But now you can see both the chromosomes are still attached. And from what they're attached from the point of crossing over. So both the chromosomes are attached to each other. मेरा chromosome ये और ये यहाँ पे जो attachment है, these two chromosomes are attached to each other from the point of crossing over. So both the chromosomes, याद रखना. So chromosomes are attached now. Chromosomes, just wait a minute. Chromosomes are attached. Chromosomes are attached. Chromosomes are attached from the point of crossing over. So where the crossing over has taken place, from that particular point, these chromosomes have been attached. Right? So they are attached from the point of crossing over. That is something you should know. They are attached from the point of your crossing over. And this point of attachment where the crossing over has taken place, the point of attachment over here, this point of attachment now has a name over here, and that name is called as my chiasmata. This one called as my chiasmata. Chiasmata or chiasmata, whatever you want to call it, this is called as my chiasmata. Can be seen. So please remember this thing very clearly over here, that this point of attachment is now only my is only my crossing over point. Both the chromosomes are attached only from the point of crossing over. Each may say from the center they are free, right? And this point of attachment now is called as my chiasmata, and this chiasmata looks like a X-shaped structure. So they have this chiasmata over here, or chiasmata, whatever you want to call it as a, and this is one, and this is one. Right? So if someone asks you that what happens in my diplotin stage, the first thing you have to tell them, there is going to be a dissolution of the synaptic complex. So from the center wala part, from the center, what will happen? The synaptic complex will dissolve. Once the synaptic complex will dissolve, what will happen? Now my chromosomes are free from the center, but they are still attached. They are attached from the point of crossing over. And that point of crossing over now will have a name, and that name is called as my chiasmata. So they will ask you a question. They will simply ask you a question that where does you get to see a chiasmata, right? Or chiasmata will be seen at which point, at which stage of my prophase 1? Your answer should be my diplotin stage.
Diplotin.
Right, hands down, diplotin stage is the stage where I'll be finding this chiasmata will be observed. So have a look over here, what has been mentioned. So you can see nucleoprotein fusion complex dissolve partially. That is your synaptic complex will start dissolving partially over here. Homologous chromosomes separate. So homologous chromosomes are going to separate except at a region of crossing over, something I've told you. And that point is called as my chiasmata. So this is something should be remembered by all of you. This is how it's going to look like. This is how it's going to look like. So you have this chiasmata at this point of crossing over, and this point as well. Right? So these are the stages we have seen right now. We have seen my diplotin stage. We have seen my pachytene before that. Before that, we have a zygotine, and before that, we have a your liptotin stage. Now we'll have the last stage, that is my dikinesis. So let me show you over here what happens in my dikinesis. So now you have the last stage, that is your dikinesis. अच्छा, how many of you will be able to come tomorrow at 5:30? कौन कौन आ सकता है 5:30 वाले क्लास के अंदर? सिखा सकती है गायत्री, possible for you to come to 5:30 वाला class? Misbah, Aisha, if possible, do come to 5:30 वाला class, because what is happening, this batch will get one lecture less. This batch will get one lecture less. So try to come in the 5:30 wala class tomorrow, because in tomorrow's class we will go beyond this. Whatever we have done today's class, now we will go beyond this in the tomorrow's class, and then you will have a test on Sunday. You won't get another class after that, no. So please try to manage for tomorrow and come in the 5:30 wala class, so that you can have a look at the things which we will do tomorrow, right? So kal aa jana, theek hai? Okay. So now let's see what we have in the dikinesis. Now let me draw a picture over here. Let me quickly draw a picture of a story of my dikinesis. Unequal size का बन गया, लेकिन ठीक है. So you have this chromosome, and now you have the green one. Now green one is now attached to the yellow one only from the tip area. Green one is attached to the yellow one only from the area of a tip. So now you can see over here what has happened over here. The point of attachment before this was my crossing over point. So what has happened? The point of attachment have shifted towards the tip. So that is the same thing I'm going to write down over here. The point of attachment has shifted towards the tip. Your point of attachment has shifted towards the tip. You can very clearly see now the point of attachment is no longer the crossing over point. It has gone towards the tip, right? क्या ये बात समझ आ रही है आपको यहाँ पे? Is this thing clear to all of you?
Yes,
Yes,
sir.
sir.
right? You should be clear with Misbah. I hope it's clear to you as well. Gayatri, Misbah, Shikha, and
Yes, sir.
who was the last person? Yeah, these are the ones. Right, and yeah. So I hope it's and Aisha. I hope it's clear to you.
Yes, sir.
Yeah. So what has been happening that the point of attachment has been shifted towards the tip, right? It has gone towards the tip over here. Now what has happened over here? Now if it has gone towards the tip, this event is called as my terminalization. When the point of attachment has shifted towards the tip, this event is called as my terminalization. This is called as my terminalization. So terminalization simply means what over here? That your point of attachment has gone towards the tip, right? Your point of attachment has been shifted towards the tip, right? This event is called as my terminalization. So they will ask you that when does the terminalization take place, then the answer is going to be my dikinesis. Dikinesis is a stage where you get to see my terminalization will happen, right? So point of attachment has shifted towards the tip. This is called as my terminalization, right? And this will take place in the dikinesis. So this event will happen, and after this you will find the prophase is now going to end. Now a few things will happen after this, like I will tell you. Now what will happen? Your nuclear membrane will disappear. Something you know happens at the end of the prophase. So nuclear membrane is going to disappear, right? So nuclear membrane is going to disappear. The second thing is going to be your nucleolus is also going to disappear. Nucleolus also disappears. Now, these are the events you find happening at the end of the prophase generally, and these are the events will happen in the end of the prophase. So here my prophase 1 is actually ending. So this is where my prophase 1 gets completed. So last stage is my dikinesis, where what you get to see, my terminalization will happen. That means my point of attachment will shift towards the tip. And after that, I will get to see that my nuclear membrane will disappear, and then you will find my nucleolus will also disappear over here. So this is what you should remember is happening during the dikinesis. So these are my 5 stages of my meiosis, or I would say these are my 5 stages of my prophase 1, and these are the stages which should be very clear to you, right? इसमें से क्वेश्चंस आते हैं, संडे के एग्जाम में भी पूछा था. The NEET exam which took place on Sunday also had couple of questions from this chapter, and one of the question was based on all these stages over here, right? So now, एक बार फिर I'll give you a quick revision before we get into the. We'll solve some questions so that I get assurance that you know all those things. So first of all, you should know you have a liptotin stage. It's a simple stage. Chromatin will get condensed from chromosome, and centrioles will start moving towards the poles. Nothing else is happening over here. Then you have a zygotin. Homologous pairing will be seen happening over here. So all the homologous pair will come together and will get themselves attached. How they will get themselves attached? With the help of my synaptic complex. Synaptic complex can be also called, it is a one type of my nucleoprotein. It can be also called as my zipper protein. Right? And once the homologous pairing has taken place, you will say my synapsis has taken place. Once the homologous pairing has taken place, you will very clearly say my synapsis has taken place. Right? After that, what you will get to see that एक बार मेरा synapsis हो गया. Then what you find that you can see that my tetrad or bivalent can be seen, means once the homologous chromosome has been formed, you can clearly see that my bivalent has been formed or my tetrad had been formed, right? Always remember a bivalent will have 2 centromere, 4 chromatid, right? And 2 chromosomes. That should be clear to you. So that is something you find happening in the zygotin stage, and now you'll have one of the important stage of the prophase 1, that's my packaging. This is where you will find crossing over and recombination will take place. But before that, what will be seen that the area where the chromosomes have been attached to each other, you will get to have a presence of a dense area will be observed. A dense area will be observed in that region, and that dense area is called as my recombination nodule. That dense area is being called as my recombination nodule, and in that recombination nodule you will get to have a presence of a recombinase enzyme. In that recombination nodules, you will have a presence of a recombinase enzyme, and this enzyme actually helps in the exchange of a genetic material. So we are doing the crossing over so that we can exchange a genetic material, and that can be done with the help of my recombinase, and this is what we'll be finding happening over here. So recombination crossing over मेरा यहाँ पे हो गया. Then you will have the diplotin stage. This is where you will get to see. The first thing you have to mention is going to be my dissolution of my synaptic complex will take place over here. Right? So that means from the center, my chromosomes are free, but they're still attached from the point of crossing over, and that point is called as my chiasmata. These are the two things you have to know in the diplotin stage. After that, you will have your dikinesis. So my point of crossing over has moved towards the tip, and my both the chromosomes over here have been attached to each other from the tip. And this event of your point of attachment going towards the tip is called as my terminalization. This is called as my terminalization, right? And once it has been done, after that we are coming to the end of the prophase 1. My nuclear membrane will disappear and my nucleolus will also disappear. This is what happens in the dikinesis. ठीक है? So अगर यहाँ तक ठीक है आप सबको, so that's something we chiasmata shift towards the end of the chromosome. This is known as my terminalization. Nuclear membrane is going to degenerate. Nucleolus is going to degenerate. Now, let's solve some questions. Question number 1, meiosis 1 results in, raise your hand to answer this. Simple question है आपका यहाँ पे. Meiosis 1 is going to result in what? बताइए मिसबा.
Production of gametes.
बस इतना ही.
नहीं, all of the above.
हाँ, all of the above. Everything is happening over here. Gamete formation, variation आ रहा है आपका यहाँ पे, and reduction in the number of chromosome. So all of the above will be the correct answer over here. ठीक है. Now, in meiosis, crossing over is initiated at. Raise your hand to answer this. Simple question है. Raise your hand to answer this quickly. All of you, crossing over is going to be initiated at which stage? All right, Gayatri, tell me what will be the answer over here? Yeah, it will take place in my packaging stage. So packaging stage is the one in which my crossing over will take place over here, right? That's true. Very good. Crossing over recombination will happen in the packaging stage. If I ask you when I'll get to see the recombination nodules, so what will be the answer? Again, packaging only. हाँ, recombination nodule, recombinase सबकी बात आपके यहाँ पे होती है in the packaging stage only, right? Arrange the following events of meiosis in the correct sequence. So now you have to look at all the events over here and then you have to arrange them. So take your time, take your 30 seconds and then raise your hand. Take your time and then tell me what how these events are going to be rearranged. Arrange the following events of meiosis in the correct sequence. So yeah, करो करो हो जाएगा. It's a simple thing. शिखा बताइए.
सर ऑप्शन ए.
पक्का?
यस सर.
शिखा ने बोला ऑप्शन ए यहाँ पे. ठीक है, गायत्री आपको क्या लग रहा है?
सर ऑप्शन ए सर. कौन सा?
ऑप्शन ए. मिसबा, आयशा आप भी बताओ, अभी ट्राई करो क्या होगा. Yes, all of you are saying option A. Yes. So it's your, let's see what is happening in option A. So you say it is going to be your synapses. Then you are saying it will be my crossing over. Then you are telling me it will be my termination of chiasmata, and then you have a difference of nucleolus. So yes, option A becomes my correct answer. Right. The enzyme recombinase is required at which stage of meiosis? Unmute करके वैसे ही बता दो इसका आंसर जोर से सब लोग. Unmute yourself and give me the answer. You don't have to raise your hand. Tell me. Question number 12.
पैकेटिंग.
Right. Packaging is going to be the correct answer. True. The complex formed by a pair of synapse homologous chromosome is called. Once the homologous chromosome forms the synapses, then what that complex is called as? Tell us, Misbah. बताइए.
Bivalent.
Right. Bivalent is going to be a correct answer. That is what my after synapses the complex is called as, right? So these are the kind of questions I want to ask you. Dissolution of synaptic complex occurs during. Raise your hand to answer this. Dissolution of synaptic complex is going to take place during. Tell us, Shikha, what will be the answer over here? Shikha is thinking. Gayatri, tell us till then. What is the answer? Again,
The diplotin, sir.
The diplotin.
diplotin. All of you are answering diplotin. Then that's a correct answer. Diplotin is the time where I'll be finding my dissociation of scientific complex will happen. So yes, that's my option C over here. These are the kind of questions I'm going to ask you. Match the following. Take your time. Take your 30 seconds and do the match the following over here. Take your 30 seconds and do the match the following. ले लो अपना टाइम ले लो. आयशा आप भी ट्राई कर सकते हो. बड़ा ही आसान क्वेश्चन है. I know you also know this. You are just not answering. Maybe Aisha, you are having your dinner right now. Achha. Yeah, Gayatri, what do you think?
So option B.
Gayatri is saying option B over here. Misbah, what do you think?
Option B.
Option B. Shikha?
Sir, option B.
Option B. So you are saying option B. All right. With A, you are saying it will go to synapses. That's true. यहाँ पे आंसर मिल गया वैसे. एक में ही आंसर मिल जाता है. At times you will find this happening that only in a single match the following single match you do and you will get your answer. So A के साथ 4 यहाँ पे आ रहा है. Then B के साथ 3. Packaging will be known for my crossing over. Diplotin is going to be known for my chiasmatic formation and diagnosis will be known for my terminalization. So yes, very good. Option B, right? So these are the few questions we have done. Now let's go back and do also those questions which we have missed, right? These are the questions we have done, right? Now let me give you, okay. Event of telophase are. So which of them are actually the event of my telophase? Now take your time and then raise your hand. Event of telophase are going to be what? A simple question. Don't you are able to answer the meiosis? You should be able to answer this. Tell us, Gayatri.
So all the above.
Okay, Shikha.
Sir, option D.
And Misbah must be also answering option D only. So yes, there's going to be a correct answer. Chromosome cluster at opposite poles and their identity is lost as a discrete unit element. So that will be happening. So they will chromosomes actually turn into your chromatin, something we have already discussed, right? Nuclear envelope assembles around the chromosome cluster. That's the second thing which will happen. And the third thing, yes, all the events are of my telophase. Right now, so this is something we have done. So over here, my prophase 1 gets completed, right? Now, once our prophase 1 gets completed, now we are going to start with my metaphase 1. So now we are going to start with my metaphase 1. Now, let me just draw a picture of a metaphase 1 first of all. So prophase 1, yaad rakhna, you have a liptotin, zygotin, packaging, diplotin, and diakinesis. This is again super important. These are the events which have been done over here. And once you're done with that, you jump into metaphase 1. After this, the story is going to be quite simple. The story from here is not going to be that difficult for you, right? And please remember meiosis, whenever it has been done, you will find it has been done during the gamete formation. It has been done during the sexual mode of reproduction. And whenever the meiosis happens, it is a reduction division. The number of chromosomes are going to decrease. So now let's come back over here and now let's understand a couple of things. We are done with everything over here, right? If you are able to understand all this thing, that means you are able to understand the major big thing. Now let's see what will happen. During the metaphase 1, during your metaphase 1, what do you find happening here is that all your chromosomes now will get, all your homologous pair will get aligned in the center. So all your homologous pair will get itself aligned in the center. Right, so let me just draw first of all green one. And then the yellow one. And they are attached to each other only from the tip. Do remember, they are still attached to each other, but only from the tip wala area. Baki beech mein se, they are all free. Right? So this is how all the homologous pair will be attached to each other only from the tip and they will all get aligned in the center. Now tell me something, inko center mein kaun leke aayega? Who will bring them in the center?
Spindle fibers.
Spindle fiber, always. That is not going to change. They will be brought into the center with the help of my spindle fiber. So you have this chromosomes over here. You have this chromosomes. Let me make a centromere over here. Right. So you have your centromeres, and they're all brought into the center with the help of my spindle fiber. Let's make some spindle fiber. And the spindle fiber are coming from where?
Centrioles.
Centrioles. The spindle fiber are coming from centrioles. They're attached to the kinetochores. Okay, so I'm not drawing over here the center, but do understand the spindle fiber are coming from the centrioles, and they are attaching themselves to the kinetochore. So all this kinetochore over here will have attachment with this particular spindle fiber. This is what you find happening in my metaphase 1. So they will ask you what is the difference between the normal metaphase of mitosis and in the metaphase 1 of the meiosis. So please remember, in the metaphase 1 of meiosis 1, you will find there's a homologous pair which is aligned in the center. The homologous pair is aligned in the center, while on the other hand, in the normal metaphase of mitosis, you don't find pairing happening. So all homologous chromosomes are aligned in the center, but they are not in pair. Over here, the homologous chromosome can be seen present over there in a pair. So this is what you have to remember in this. That is what you find happening in my metaphase 1. ठीक है? तो ये बात आप भूलना मत, ये चीज आपको यहाँ पे आप सबको यहाँ पे ऐड रखनी है. So this is something should be clear for you, right? So I hope this is clear to you. There is something clear to everyone ना यहाँ पे?
Yes, sir.
This is what you find happening in my metaphase 1 के अंदर आपको ये चीज आपको होते हुए आप यहाँ पे मिल जाती है. This is what you will get to see happening, right? After that, you will have the anaphase 1. Anaphase 1 we'll discuss tomorrow. Please make sure that all 3 of you, 4 of you, please do join the class tomorrow and do join this 5:30 wala class, right? Because as I've told you also that we might not get one more lecture after this, right? You will directly have a test on Sunday. So do join tomorrow's 5:30 wala class so that whatever we'll be doing tomorrow, we'll be doing beyond this. So in that batch also we have done till this, and we'll be going beyond this. We'll be solving number of questions, and we'll be solving mainly questions, and we'll be completing the meiosis, which is now it's not a big thing to do. Yeah, we'll be doing one important thing tomorrow, which is going to be your cell cytokinesis in the plant cell, right? So please make sure you don't miss out tomorrow's class, right? So Shikha, Misbah, and Gayatri, do come in Baji class tomorrow at 5:30, right? फोन नहीं है तो फोन ले लेना अपने लाइक सिस्टर या भाई बहन से कि यार एक दिन के लिए चाहिए, ठीक है? But to come in the tomorrow's 5:30 wala class, right? So yeah, this is what we have done. And if you are able to solve this question, so I will share with you 2 assignments right now. What you have to do now, you have to solve the assignment on a piece of paper and share that with me. Like take a photograph. You don't have to write a complete question and the answer. You just have to write question number 1, answer B, question number 2, answer C. Take a picture of that and share that with me, right? So that I will share that with you the answer key. You will only get 20 questions, which won't take more amount of time. ठीक है? So वो चीज का आप यहाँ पे ध्यान रखना. I hope the instructions are clear to you.
Yes, sir.
Yes, sir.
Yeah. All right. Now before we wrap up the class, so I hope the pace of the class is okay. I hope you are able to understand everything, whatever we have been taught at a certain pace.
Yes, sir.
Yes, sir.
Language, I hope it's not a concern. Misbah, Gayatri, I hope language is not a concern.
Yes, sir. Yes, sir.
Mm-hmm.